Welcome back. Let's continue to look at applications of network biology. There are a lot of different kinds of applications of network biology. Some of them are focused on looking at important nodes in the network, important edges in the network, and so on. And others are focused on trying to make biological inferences or biological predictions. In fact, networks are essentially models, right? Models that help you understand the system better. So we looked at different kinds of models of real world networks in terms of they could be small world networks, power law networks, and so on. So all of these models are intended to help us understand how networks function. Right? So today what we are going to look at is a few applications of network biology. Let us start with what is known as centrality lethality. So this is essentially known as the centrality lethality hypothesis, which states that the more central nodes in a network are more likely to be essential for biological function. And this stems from a very classic paper in 2001, which is what we are going to talk about briefly today. So this is a classic paper in Nature in 2001, and you might have seen this picture a lot. I would have shown you a few times already. So what is seen here is the largest cluster containing 78% of all proteins in yeast. So this looks at a yeast protein-protein interaction network. I have already taught you how these kinds of interaction networks can be constructed. There are interaction networks and functional association networks that we have talked about. But what we see here is a yeast protein-protein physical interaction network, right? Put physical in the brackets. So protein-protein physical interaction network, wherein these are proteins that are known to bind with one another in yeast. And these have been identified by high throughput experiments and so on. And now you take every pair of proteins that bind or interact with each other and you connect them by an edge. And what you get now is a network, a protein interaction network that has as many nodes as proteins in yeast. And now you can imagine that it is possible that some of these nodes may be disconnected, from a large network even, right? We've looked at the concept of connected components and so on. So one fraction of nodes, a large fraction of nodes typically sits in the largest connected component. This is very common in all biological networks. You will see that if you perform any analysis of these kinds of networks in any of your assignments, you will notice that a very large fraction of nodes sit in a single connected component in the network. And that is what is the component that is shown on your screen right now. So this is the largest cluster containing about 78% of all proteins, and the color of each node signifies what happens when you remove this node from yeast, not from the network. You actually perform an experiment to knock this protein off from yeast and study what is the effect of this removal. So if the node is colored red, it means that the organism, the yeast, dies following the removal of this particular protein from the cell, right? And if it is green, it means that it is non-lethal. If it is orange, it means it is a slow grower, meaning the cell survives, but its growth rate is much lower than what it was originally. And you also have these yellow nodes, which probably represent failed experiments or where the outcome wasn't very obviously clear and so on. So you have four different kinds of phenotypes that are captured here: lethal, non-lethal, slow growth, and unknown. So these four colors are what you see in this protein interaction network on your. What is now interesting is that what are the patterns that you see? What kind of proteins are more likely to be essential? What kind of proteins are less likely to be essential, and so on? And this is what Jeong and others found. They saw that although proteins with less than 5 links constitute about 93%, can you already start guessing what kind of a degree distribution this looks like? So you have 93% of the nodes which have 5% of the links. You have 93% of the nodes which have less than 5 links, and you have 0.7% of the nodes with over 15 links. So this is a classic case of a power law distribution where you have a really skewed distribution. You have a long tail where you have all of these proteins with greater than 15 links, and you have the big spike that you see in the close to the origin, right, where you have a lot of proteins with 93% of the proteins with less than equal to 5 links. But it turns out that of these proteins, barely 1/5 of them were essential, of the proteins with very, very low connectivity. On the other hand, only 0.7% of the proteins have greater than 15 links. What are these? These are your hubs, remember. But a single deletion of 62% of them proves lethal. And so they say something like this. I am not very fond of how they present it, but they say that proteins, highly connected proteins with a central role in the network architecture, are 3 times more likely to be essential than proteins with only a small number of links. But the point here is well taken. The point is, the more central a protein, and they are looking only at degree centrality, remember. We could certainly look at other centralities. I will talk about that in a moment. So proteins that have very high degree centrality are very, very likely to be lethal in the context of yeast's protein interaction network. And there is another important concept to note here. This is a concept that we haven't dealt with in great detail, but there is this notion of what is known as assortativity. Or also known as homophily. What does this mean? So, homophily, as the name sounds, is homo means similar and phily means liking. So, there is a liking for similar nodes or a tendency to connect with similar nodes. And what do we mean by similar here? Here we talk about essentiality as a key defining feature, but you could talk about degree, you could talk about other attributes of a node and so on. Right. So, in a regular social network, you can see if people with the same interests connect to each other, which is what typically happens, right. People who like particular kinds of sports, people who kind particular kinds of movies or whatever, typically tend to be friends, right. And so, this is called assortative mixing, meaning you have far more connections between nodes that have between nodes that are similar in nature. On the other hand, in biology, you typically find disassortative mixing, wherein here we are looking at nodes in terms of degree. So, high degree nodes typically connect with low degree nodes, or also essential nodes typically connect with non-essential nodes. And this is something that you find on a regular basis in biological networks, and this is quite interesting. So, how do you quantitate assortativity? Newman has a measure. I would invite you to go and read about it. So, Newman's measure essentially tries to quantitate what is the fraction of nodes, I mean how likely are you to connect with nodes that are of the same nature than by chance. Right? If 99% of the nodes in a network are of a particular type, right, are say non-essential, and 1% of the nodes are essential, it is very likely that a non-essential node is going to connect to a non-essential node. Right, and an essential node is also likely to connect to a non-essential node, but is this assortative? And that is where you need to build a good control and build an expectation and try to identify if the connection between essential and essential or non-essential and non-essential are much higher than you would expect by chance for this kind of a distribution. You expect at least a heavy amount of connection between essential and non-essential in this kind of a skewed nature of node types. And do you find more than that or less than that? That becomes the question which decides how assortative the mixing in the network it is. So, this was captured in a very nice paper by Newman in 2003 called Mixing Patterns in Networks. So, predicting essential genes. So, now can you use this information to predict essential genes? We already did a study in 2014 wherein we looked at different measures of centrality. We looked at degree centrality and betweenness centrality in terms of their ability to correlate essentially with the centrality lethality hypothesis. So, our paper is called Revisiting the Centrality Lethality Hypothesis, where we try to identify some of these structural determinants of the network and how they influence what if network structure and network position can actually influence the lethality of a particular gene in the network. But we extended that further here to actually go ahead and predict essential genes in a number of organisms for which this data is actually available. There is essentiality data available for a bunch of organisms, at least 27 organisms, or maybe I think it's 31, but only some of them had networks available. So this is in the database of essential genes. So we'll see what is our approach. The question is, can we predict essential genes across organisms? Can you learn patterns? And so one way is to simply say that centrality is lethality. Higher the centrality, higher the lethality. But that's a sweeping statement, and it's not necessarily a very correct one, because it's possible that you find a highly connected node which is non-lethal, simply because if that node are duplicated, you would have another node with very similar profile, similar connectivity, both nodes with very high connectivity, but neither of them are going to be lethal, because if one is damaged or one is inhibited, the other one can kick in and rescue. So this kind of redundancy exists in biological networks. So it is not necessarily that degree centrality alone plays a role, right? There are other centrality measures which may also play a role, and there are these kinds of duplication events and so on, which further complicate the playing field for us to make reliable predictions. So how do we go about this? Today we have a large number of organisms for which we have genomes and interactomes available, and we've already studied about that, the string database and so on, and you have huge networks of functional associations in an extraordinary number of organisms. And can we ask the question, does protein function or essentiality depend on network position or parameters? And remember, the centrality lethality hypothesis, in fact we should call it the degree centrality lethality hypothesis, is still a hypothesis, and there are challenges, right? So it need not, as I was mentioning earlier on, you could have a protein with very high centrality that's not at all lethal. So this correlation is strong but not absolute. So can we leverage this network information that is available in droves in databases such as the string to predict essential genes in a reliable fashion? And we used data from the string, and our approach was, we used a combination of network features that capture global, local and neighbourhood features. We used a bunch of centrality measures that I will mention shortly, but we also used these metrics known as refx and rolx, which are basically, which stands for recursive feature extraction and so on, which tries to extract a lot of features from a network, for a node from a network based on its neighboring nodes and so on. Right? So it recursively, for example, if a node has a particular property and its neighbors has a particular property, those neighbor properties are also added to this node in a recursive fashion. Right? So you really understand things at a slightly more global level, or like semi-local or neighborhood level. Right? So let's just stop for a moment and ask ourselves, what is global, local and neighborhood properties? Right? So when you look at a single node and identify its properties, right? So you look at its degree or so on, you're very local, because this is not immediately or easily affected by the rest of the nodes in the network. But when you look at something like betweenness centrality itself is conditioned on the number of paths in the network and so on. So this captures something more than a merely local phenomenon or a mere immediate neighborhood of a protein or a node in a network. Right? So betweenness centrality is a slightly more global measure. Of course, things like average degree or so are more global measures, and local, you can think of things like random walk centrality. Right? A random walk centrality, with as you tune the alpha, if you make alpha like if you make alpha very low, it means that you will not reset the walk very easily, right? So you will explore larger and larger regions around a particular node. So what you see is that, but when you have alpha like very high, you are basically looking at the immediate neighborhood, right? So with a very high probability, you will reset the random walk and restart from the existing, restart from the starting node again and again. So these are ways to try and capture different aspects of a network by starting at a particular node. Right? How much of the network are you seeing around this node, and what implication does it have for the structure of the network, navigability of the network, and in this case, can you use that to predict essential genes or important lethal proteins in a network and so on. So the data that we used are, for the essentiality data we used the database of essential genes version 15.2. It had data on 31 organisms, out of which 27 had interactomes in the string. We used string version 9.1, and string has since been updated. You have many new versions which have more and more features and more algorithms that capture various kinds of associations. We already talked about some of these functional association approaches a little earlier. And there are 12 centrality measures that we found highly useful. What were these? Many of these are familiar to you, and those that aren't, I invite you to go and read a little bit more about them. So, closeness centrality, betweenness centrality, degree centrality, eigenvector centrality, subgraph centrality, information centrality, random walk betweenness centrality, load centrality, harmonic centrality, reaching centrality, edge clustering coefficient centrality, and of course the famous PageRank. So, we used these metrics and a few other metrics as well in addition, and we made different feature sets. I'll show you in a future slide. And we also performed a sort of validation by leaving one species out to ensure that cross validation of a sort, to ensure that we are not overfitting the model too much and so on. It's a challenge given that there is so little data available. So, what were our challenges? One of our challenges was class imbalance. Only 11% of genes were essential on average, and in certain organisms it was as low as 2%. So, this becomes very challenging, which means that you must have already studied about class imbalance, but let me just quickly make a remark about it. So, when you have class imbalance, what you see is that you can easily predict everything as the majority class and still survive with a very small error, right. Your recall suffers, but your precision is actually quite reasonable. So, if I predicted every gene in Pseudomonas aeruginosa is non-essential, right, I will have an accuracy of 98%, but then I will have a recall of 0%. So, this is the challenge that we have to grapple with. So, how do you tune and how do you make sure that you train the classifier to actually understand this issue and make predictions that are more reliable. And we also had a huge number of features. So, feature selection was a challenge. How do you pick the right features from as many as 283 network features or another 200 odd sequence features as well. So, these are all features that are readily generatable given a sequence or given a network. So, it's quite easy to produce these for any organism of your interest. And what was our approach? We used SVMs for classification and we used lasso for feature selection, and we also handled class imbalance by resampling the minority class. And we also did leave one species out for validation. And this is what our final results look like. Liu et al. Was a classic paper that was published a few years before our work, and you see that all of our approaches actually outperformed it significantly, both in terms of all the metrics, right? Either if you look at AUROC or the precision or recall, and you see that some of our approaches were like really strong. And when we looked at the top 200 odd network structure, top 200 odd features, we got a fairly good performance, right. So, almost an 86% AUROC and a fairly good precision and recall. So, to summarize, we engineered several potent network based features for predicting gene essentiality across organisms. Right, it is easier to predict these in a single organism than across organisms, which means that you have to generalize a lot more, and even a few features like 12 features can achieve a very high accuracy for classification. As you saw in the previous slide, with just 12 features we are able to get a fairly good performance, which beats what was the then state of the art. And augmenting network based features with sequence based features afforded just a marginal improvement in performance, so nothing to write home about that. And our proposed set of features can be derived for any organism containing both sequence and interactome information, such as those in the string. So, we can readily apply it on all string organisms, which is what we did, and I will show you shortly. And you do not need to really go for sequence as well, because we said 12 network features are more than sufficient. Right. So, I would like to acknowledge Karthik and Ravi who played a major role in this study. And you can see all our codes from GitHub, and a lot of our data is accessible from there. Okay. So, we then pulled all of this together in a very nice database called NetGenes, which predicts essential genes for 2700 plus bacterial interactomes from string. So, pulling these string interactomes, we compute all these centrality metrics and we build a classifier. We perform the feature extraction and we build the classifier, and we provide essentiality scores, annotations, and also the feature vectors if somebody wants to try and build a better classifier and so on, and our database is available from NetGenes, and our database is available from this URL. So, this is how NetGenes looks like. This was built by Sinha and in collaboration with Professor Ravindran. And what did we do? We have this very nice database which has 2700 plus organisms, and you can just download the essential genes for each of these organisms directly. It's got a very clean and quick interface which is easy to access. With that, we've now looked at predicting essentiality from networks. Right, we already looked at the centrality lethality hypothesis, which mostly focuses on just degree centrality as an important feature for predicting lethality. But now when you combine it with several other network features, you are able to make a much better prediction as we showed here. And this algorithm that was originally developed in 2018, we now also have a database which helps you look at all the essential genes in string interactomes. So, with that we sort of come to the end of this particular application. We now move on to a different application.